precious cell pellet with my protein inside. But from the bubbling biochemists, those proteins cannot hide. So there are proteins inside of here that I want to purify. So I put the instructions for making the proteins into insect cells and got the insect cells to make them for me. I'm using this baculovirus system. I have more on another post. But I can use, there are techniques that I can use to purify out the protein. So I can use like protein chromatography methods where I have columns filled with little beads that interact with different proteins differently so you can separate the proteins, and I have more on that in other posts. But in order to do all of that, I basically need to first get them outside of these cells and separate them from all the cell membrane and the DNA and all that other stuff, so I can just be separating those proteins using the chromatography methods. So today I want to walk you through how we do that first part as I do it. So we basically need to thaw the cells, so I'm going to stick them in this water bath, and so for bacterial cells, you can put the water bath higher, but because they grow at like 37 and stuff. But for the insect cells, I'm setting it at 27, and then I am going to add high salt to kind of disrupt the membranes, then do ultrasonication to really break them open and help shear up the DNA, so basically tear it into pieces. Then I'm going to add PEI, which is this cationic polymer, so it's positively charged, DNA is negatively charged, so it's going to neutralize the DNA and help it like pellet out, or not pellet out, but precipitate out. And then what I'm going to do is I'm going to spin them really, really fast in the ultracentrifuge, and this is going to now take all those membrane bits and the DNA bits and any other insoluble gunk, and that stuff's going to be heavier, so it's going to sink to the bottom. And then all the protein and salts and various things are going to be in this liquid part, the supernatant. And then now that I have the supernatant, I can just like syringe filter it to get rid of any big things that are going to clog up my chromatography, and then it's onto the chromatography. So today I'm just going to give you an overview about how these things work, and some technical tips and advice and practical stuff as I do one of my closest to last purifications in this lab before starting my postdoc. And so yeah, so let's take a look. The actual conditions and everything are going to vary from protein prep to protein prep depending on how much of the protein you have. So like this is a small scale expression, this is just a V1, so just like an initial small scale thing for insect cell expression that you're using typically just to make more virus to infect more and more cells, more than that in the insect one. But basically the point is that it's a lot less volume than I'm normally working with, a lot less protein than I'm normally working with, so I'm like scaling things down a little. And so that's one thing that matters. The other thing that matters is like what your protein conditions. Some proteins will tolerate higher salts than others, some lower salts than others. The other thing is the type of cells, and so this is going to be for insect cells. Insect cells are easier to break open than bacterial cells, so bacterial cells are going to take a little more work, so a little more on that in the sonication post. In the sonication part of the video, I mean a little more, basically because they have cell walls and stuff too, they're harder to break open, and so that's where that part will come in. But once you break them open, then they're actually like easier. There's like a thinner lysate and stuff to work with than the insect cells, so we can actually syringe filter them through a smaller pored membrane. And so you want to filter them through the smallest pore you can find, but you don't want to, it needs to actually be able to go through, and without like, yeah, it can be hard to push things through. But yeah, so the basic overview is going to vary from cell type to cell type and that sort of thing. There are other ways to lyse cells as well, and so I'm going to show you, I use this like salt and sonication method, but there are other methods. You'll see a lot of times with people doing like cell biology type of work, say maybe they just want to see what's in a protein, they do like Western blots and that sort of thing. They just have little small samples of cells and that sort of thing, and you typically often use a lysis buffer that has a detergent in it, so like a soap, to help break up all those membranes. When we're doing the protein purification, we typically don't want detergent unless we're doing something with like membrane proteins or that sort of thing, because we don't want it to interfere with anything. We don't want it to be solubilizing the membranes that we're trying to get to pellet out. So instead of using detergent, we use these other more mechanical methods. There are also enzymes like benzonase nuclease, various enzymes that you can use to help with the lysis, especially for bacterial cells that, as I mentioned, were more difficult to break open. And so there are also other methods using like beads. There's the thing called a French press, where you have a high pressure and you're going to force the things through like a fine needle, and shear things that way and break things open that way. But I use the Sonication method, where basically you're making little bubbles, and then the bubbles are crashing, and it's making energy, and it's making low and high pressure, and then in the low pressure zones, you have the energy and you have the low pressure, so bubbles can form. Then the high pressure zone comes, gets those bubbles to collapse, and voila, you have like energy shocks, and then that makes things break open, and it makes the DNA shear apart, and that. So I check these periodically to see when they're thawed, and they look like they're thawed now. And so now what I'm going to do is I'm going to pour them into a little beaker and then add salt. So I'm going to use buffers that are have potassium chloride. So I'm going to add potassium chloride, 1 to 10 dilution of the KCl. So I'm adding about an additional 300 millimolar worth, and so I have about 15 mils here. And I am going to add 1.5 mils of the KCl, just using like a pipette man. Then I'll swirl it around a little and take it off to the sonicator. So this next part of the video, it was actually I made it a while back and last year. So when I walk you through the actual ultrasonication part, and then I will meet you back on the other side. Sonicate and lyse, come on! Protein purifier, sonicate. So right now I am starting a protein purification. So I had bacteria express a protein make a protein that I'm interested in by putting the genetic recipe for it into the bacteria, as we talked about before. And so these are the cells that after they made the protein. And right now my job is to break them open and purify the protein out. So to help me with the break them open part, and to also shear the DNA, as I'll talk about, so basically shred up the DNA so it's not going to interfere with my protein purifying because it gets all gloopy, I'm using this thing called an ultrasonicator. This is a probe tip ultrasonicator, so you see this probe here, and I have my, the process is, it basically works by making all these little bubbles in the liquid, and those bubbles burst, and then that generates these shock waves that break break things and stuff. And I'll talk more about it, but first I want to show you it because it's kind of interesting, and because I want to get it started. But the process is going to generate heat. So I have it in this like ice water bath that is made out of styrofoam. So it's like a styrofoam box, and then it's filled with an ice water bath, and then there's a little cutout so that your beaker doesn't like fall down, because that can happen as the ice melts because it gets hot. But basically you don't want your protein to get hot, which is why the ice. So it doesn't, there's like a little stand thing doesn't go to the right height, so we have some styrofoam blocks on top. But what I'm going to do is I'm going to hook it up and put the probe inside, and then I'm going to set it to go for bursts. So it's like 2 seconds on, 4 seconds off to go to second, and then it just keeps going. So I do several cycles of that to help with it. Yeah, so that's the probe sonicator. We also have like a bath sonicator that you might remember from me cleaning the cryolids from the crystals off. So it's kind of like this. And you might have also have some sort of sonicator that you've used to clean your jewelry. So let's talk about how all this works. Quick note that I'm not a physicist, I'm a biochemist, so I'm doing my best, and hopefully I get the terminology right. So basically it's ultrasound waves. They're a type of sound waves. And unlike light waves, sound wave actually has to like travel through a media. So it travels through something. So you have these waves that are actually like pushing matter, and so like pushing the molecules. And when the ultrasonicator does this, what it's doing is, so you have this wave, and so you're generating this like area of high pressure, and then area of low pressure, and high pressure and low pressure, and high pressure and low pressure. So what happens is that you have this, when you stick the tip into water, or if you're using a bath sonicator, it's coming out from the walls or something. But the sonication waves are going to travel through the water, and they're going to push those water molecules. So the water molecules right now are in a liquid form.
So basically, your buffer is going to be some water-based buffer. So we're just going to talk in terms of water. Water molecules are really sticky. They have this partially positive oxygen and this partly negative hydrogens. And so they like each other and they're going to stick together. And so you need energy in order to unstick. And so if they don't have enough energy to unstick from one another, then they're a solid. They can only kind of vibrate in place. If they have a little more energy, they can slip around one another, but every time that they break free from one of those attractions, there's another one that can hook them in. And if they have finally have enough energy, then they can be break free and be a gas. And molecules really like to be a gas because they have a lot of entropy. It's like freedom or disorder, like they can move around how they want. But they face problems in order to do this. So in the liquid, the water molecules, they're sticking together through these like intermolecular forces. So between the water molecules, these attractions. So they can, and they're facing this pressure from around them. So water molecules can kind of like form these bubbles inside. And so typically we think about this happening when you have like boiling, and that's because they all have enough energy. So energy in the form of heat, that they can then like kind of team up and escape. So when you have the water molecules, like trapped molecules in a liquid as these gaseous molecules, they're kind of like in this bubble, like, so they have water all around them, and then they're the gas, and they're kind of like pushing on the wall. So they're putting on this vapor pressure. But then they have things to deal with. They have this hydrostatic pressure from above, they have atmospheric pressure, and they have surface tension. So basically the liquid water molecules around this bubble. And so the pressure, in order to kind of be able to counteract these things, they need to have enough force to push back against all these forces. And so one of the external pressures, if we control the external pressure, if we lower the external pressure, then it's easier for these bubbles to form. But if we were to then raise the pressure, then they would collapse back down. And this is the idea with ultrasonication. So we give them energy in order to form a bubble, but they only have enough energy to counterbalance the opposing forces in the regions of low pressure. So we use these high energy waves, these ultrasonic waves. What they're going to do is they're going to move through the liquid. When they're moving through the liquid, they're actually pushing the water molecules apart, and they're like, it's like a wave. So you're getting these areas of like high pressure, low pressure, high pressure, low pressure, high pressure, low pressure. So you're both giving them energy through these waves, the water energy through these waves, and also you're creating these zones of low pressure where it's easier if they have enough energy for them to form a bubble. So you're hitting the jackpot in these zones, and these bubbles can form. But because you have this wave-like nature of these ultrasound waves, now the high pressure zone is going to come in. When they have the high pressure, now they can't keep up they don't have enough force to counterbalance the pressure, and so those bubbles are going to collapse. So they collapse in this process called gaseous cavitation, and when they do this, they're going to give off energy as these shock waves, and these shock waves are going to give enough energy to do things like help break open the cells and shear apart the strands of DNA so that they'll get less lumpy. So in order to help precipitate out that DNA that we just sheared up, we can add polyethylenamine, or PEI. And so we've seen it before as a cationic carrier, so cationic, positively charged. And if we remember, DNA is negatively charged. It's commonly used as part of the transfection reagent. And basically what it does is it'll help neutralize the backbone of the DNA so that it can get close in contact with the negatively charged phospholipids of a cell in order to help it get in. But here we're using it to precipitate the DNA. And the reason we can do this is because things are typically least soluble in their neutral form. So if you were to, so water has those partial positive, partial negative charges. And so if you have something neutral, then it's not going to offer the water anything fun. And so water tends to exclude these things and push them together in this like hydrophobic effect. So DNA is going to be least soluble when it's neutral, and it's going to be neutral when you add something positively charged to cancel out its negative charge, because it's got that negatively charged phosphate backbone. And so in this way, you're able to add PEI in order to precipitate the DNA. A word of caution though, that thing about molecules being least soluble when they're neutral, that holds true for proteins too. And so proteins are typically least soluble when they're neutral. They don't offer the water or anything to hang out with, at least on net. And so PEI is actually a way that people can use to selectively precipitate out proteins based on their charge. And so if you have a negatively charged protein and then you add a ton of PEI, you can actually end up precipitating out your protein. And at this stage, you definitely don't want that. So just be cautious and don't use too much PEI. So typically I use like 0.2% final. But you might need to play around to find the optimal conditions for your protein and your mixture.
So I have a 10% PEI solution. I'm going to do a 1 to 50 dilution, so I have a final of about 0.2%. And I am just going to add it dropwise. And you'll see as you're adding it, it's going to thicken, get like whiter as the DNA is precipitating. Sometimes it even gets harder to spin, and you'll see it'll like stop spinning and then it'll start and it goes really fast. But here, since I don't have that much, you're not able to see it as well. Then what I'm going to do is I'm going to take a big magnet and stick it on to here and pour this lysate out into an ultracentrifuge tube, which I'm going to label because I'm doing 2 preps. It's really, really important to keep things labeled. So you'll see I have numbers on everything so that I can keep track of which one is which. But yeah, so this magnet trip will help you keep the other magnet from falling out. These centrifuge tubes, you want to fill them at least halfway full, but not so that they're overflowing. Like when it's in the angle of the rotor, you don't want the liquid to be in touch with the ring. You really don't want stuff to leak out. It can cause problems with the centrifuge. And you want to make sure, so I'm going to balance these really, really well. You want to have them really balanced. It's not okay to just like eyeball guess these. It needs to be within like about 0.05 grams of one another. So I'll balance them and then I'll transfer between them in order to get it super duper balanced, because this is going to spin really, really fast. The other thing is you want to make sure you have O-rings on all of the lids and everything, so nothing leaks out. And so I actually keep your leftover lysate, and then you can just add it in. So zero it or tare it or whatever to the one that was heavier, and then just add in until you get to what you need. And be sure that you include the lids on here when you're doing this. Sometimes the lids can have slightly different weights, and that can influence things. Time to go into the ultracentrifuge, and so you note that we have this little tool so we can use to kind of like take the tubes out, because it's really hard once they're stuck in there. But this is going to spin it really, really fast. We keep these in the we keep these rotors in the fridge so that they're already cold, and then when we're running this, we'll run it at 4 degrees Celsius. So I have it set to go for half an hour at 35,000 RPM. And so this is really, really, really fast. So put it in, make sure it's all aligned properly, and then you start. And you want to make sure that it's going to pick up speed. So you see this, like the vent or whatever, it's going to like create this vacuum. And so as this should start going down once, and then once this gets down, once you have this like vacuum established, then it'll start like ramping up the speed really high. So typically it gets stuck around like 3,000 for a while, and then once it starts like ramping up above that, and once this number starts going down, then it's normally, you know, that it's going, the run is like everything's going okay. There could be problems if it can't get up to speed before then. So sometimes I stay and just watch it sometimes, but usually I come back in a minute and see if there's any problems, and there's like a red error or anything. And we also have like a log sheet that we keep track of how much we use it and if there are any problems. I think so. Even if you set it for like half an hour, it's going to be longer than half an hour. Yeah. Once it starts below like 3,000 or so, you can like open the vacuum so it stops doing that. Then it'll make a nice sound when it's done. Maybe. There was that nice sound. So when you take it out, so you can use your handy little tool to take it out, and you'll see the pellet, and I'm gonna pour it into the beakers, clean beakers I have waiting. So I have my supernatant, which is my lysate, the supernatant, and then the pellet, which has like the membrane, gunk, and stuff that I don't want. And so now I just want to syringe filter them, and then put them onto my columns. You just have to suck it up, the liquid, and then this just screws onto here, and then you push it through. I'm gonna take this all to my least favorite place, the cold room. So I'm collecting the flow through, in case the protein is in there. I also keep the pellet and the lysate. If there's problems expressing, I can see if it went to the pellet or like it's insoluble in inclusion bodies or that sort of thing. But hopefully it should be in the liquid part, and it should be sticking to the beads, and everything else should be flowing through, and then I'll wash it. And yeah, I just watch and wait and then wash, and then get my protein off. And so this is all just chromatography stuff from here on, and so I'm not going to go into it in today's post, but yeah, so hope that helped give you insight, a look to a typical, or I guess there's no real typical, but one way of getting your protein ready to go onto these columns in the first place. Oh, and of course there's the cleanup.